So today we're going to talk about the coronavirus pandemic. And I think if this cloud has a silver lining, the silver lining is that everybody gets to learn immunology 101. And I get to teach it. And so this is a good thing because the immune system is really important to keep us all together. And it wasn't until really until the AIDS epidemic, and of course the HIV infects the very T cells that are the major players in the immune system, the so-called CD4 positive helper T cells, and then they start dropping off, and then you become immunodeficient. And so that's part of the acronym for AIDS, the acquired immunodeficiency syndrome. And it wasn't until then, that people even were worried about the immune system. I can remember going to cocktail parties in New York, and people would ask me, what do you do? And I'd say, well, I'm an immunologist. And I could see them then looking over my shoulder to see who they could run off to. They didn't have to talk to this crazy, nerdy scientist anymore. And that changed with the AIDS epidemic. So people became interested, but now it's dropped off and now I think the interesting part about the whole coronavirus thing is that almost, daily you're learning virology and you're learning the immune response to that virus in spades. And it's been very, very interesting for somebody like me. And it's not very often you have a worldwide epidemic like this. And you have to bring into all of the things that you think about and talk about it. So I've been waiting for some really good papers to appear in terms of recent science done on this issue. And so recently there have been some papers out that I thought were very, very important, and I tweeted about them and repeatedly really to try to get people to read these papers because I think they're so important. And it's very interesting because back in the day, in February and early March, when I finally decided that I was going to self-quarantine at that point in time, and my wife and I underwent self-quarantine on the 8th of March, just a week before the Ides of March, and we've been self-quarantined ever since. But when I did that, or before I did that, I went to the literature and I started looking for papers. I couldn't understand, early on they were saying that the asymptomatic so-called incubation period, and all these viral infections, all infections have sort of an incubation period where people are asymptomatic from the time that they are first infected until the time they develop symptoms. That's usually just a few days, 3 or 4 or 5 days. And they started saying in the COVID-2 infection that it was like 12 to 14 days. I said, well, that's bizarre. Why is that? And the other thing they came up with about the same time, or I became aware of, is they started saying that asymptomatic people could be shedding virus. And I said to myself, well, that's why this thing is so contagious, because it seems to be ripping along. This was back when Wuhan was going crazy, and then Italy right thereafter. And it seemed like it was really contagious, much more so than any other viral infection. I guess with the exception of smallpox and measles that are also both transmitted through aerosols, so that you breathe them in and it goes into the lungs and then throughout the body, and those two viral infections. And so, but usually, as soon as the immune system starts to be working against these invaders that have come into your body, it keeps viral replication low. Usually asymptomatic people do not really shed enough virus. I guess the virus hasn't replicated to the point where they've got a lot of virus that they're giving off into the environment. And so I went to the literature and I found a really interesting review that was absolutely prescient, published in Seminars in Immunology. It was submitted in August of 2019. So it was before anybody knew anything about SARS-CoV-2 and COVID-19. And it was the senior author, the leader of the lab, was a woman by the name of Eleanor from Toronto. And it turns out Toronto, if you probably know your history now, Toronto got hit hard with the SARS-CoV-1 epidemic back in 2003. And so she, Eleanor, and I really recommend everybody look up Eleanor Fish, and because she's a hot ticket and she's really right on the money, essentially, with regard to this stuff. She published a review in Seminars in Immunology that she submitted in August of 2019, and it was published soon thereafter. Because I accessed it in like January, February, or March of 2020, and I was really surprised because what she goes through the whole biology and immunology of viral infections, and so she covers the waterfront. It's not just about coronaviruses. And she then goes into the whole biology and pathophysiology, really, of interferons, what they are and how they work, and so forth. And it's a complicated story. It's even more complicated than interleukins. And we sort of borrowed the inter from the interferons so that confused everybody. But interferons were first discovered in 1957, and I think I've gone over them in some of my immunology videos. In 1957, a group in Switzerland actually discovered that if you took some cells in a tissue culture and you infected them with a virus, and waited a day or two, and then took the supernatants from those infected cultures and culture fluids and put them onto fresh cells, and then added the virus, that would interfere with the infection of the second culture with the virus. And that became interferon, and it took a long time, more than 30 years, to really figure out what the molecule was. And it turns out that there's many different molecules that have interferon activity. And they're classified according to what receptors that they bind to on the cell surface, because that's how it works. Because interferons really became then the first so-called cytokines, of which the interleukins are part of that story. And now we've got 43 or so of the interleukins. We've also got all the hematopoietic growth factors, erythropoietin and so forth. And so there's a lot of cytokines nowadays. They all work in a very similar fashion. They all are secreted by different cells for different reasons, and they do their work by binding on to receptors on the surface of other cells, and those cells then become, in the parlance of the scientists, those are the target cells for that particular molecule. So interferons have been classified according to which receptors they bind to, because there's fewer receptors than there are interferons, because some receptors are used by a dozen or so. Type 1, type 2, type 3, and so forth. And type 1s are the ones that were first discovered and described and so forth. They are made by primarily white blood cells, primarily macrophages, and also every cell in the body has really the capacity to make interferons. And the fibroblasts were, because the scientists were growing them in culture, and that's what the virologists used, was the other source of these type 1 interferons. And what she points out in her, Eleanor Fish, what she points out in her review is that most of the viral infections that are known to man, these viruses have figured out ways to get around this interferon business. You know, we're back to Darwin again. They've co-evolved with the immune systems of all the different kinds of animals and so forth that are trying to prevent them from coming into their body and replicating. And so it turns out that interferons, as a class of cytokines, form probably the most important part of the so-called innate immune system. We have two major immune systems. One that we call innate, and then if there's nothing you have to do, it's already there. You're born with this immune system. And the other one is called the adaptive immune system, and that's the one that is usually participates later on. It takes a week or two for it to rev up and get going. So the innate immune system is really important for viral infections, because what happens is that the mammalian immune system that we're dealing with here, it's evolved ways of recognizing these viruses. And there's a whole raft of receptors that the immune system has, and the receptors on the surface of the cell, receptors on the inside of the cell. If the virus gets in there, so that the cell can say, oh, this is not a good thing. And as soon as those receptors are activated, they then program to activate the cell to make interferons. And then the interferons are secreted out into the environment and so forth. And the interferons then bind to their receptors. It gets to be redundant after a while. You have so many molecules and receptors and so forth, but they bind to their receptors and they activate a very characteristic set of molecules on the inside of the cell that then transfer the signal from the membrane, where the interferon receptors are, all the way to the nucleus, and there is a raft of interferon stimulated genes that start to be transcribed and translated, and new proteins are made, and those new proteins then block viral replication. That's good, and everything should work fine, except for the fact that the SARS-CoV-1 and then the MERS virus and the Middle East respiratory syndrome virus were found to, and this is what she says in her review article that was submitted in August of 19, is that there are several different gene products inside the virus that code for molecules that block interferon production. So they stop it right at the beginning. Can't make any interferon. And consequently, if you then fast forward to SARS-CoV-2 and the recent pandemic, what explains the fact that there is this, number one, a prolonged incubation period, and number two, the fact that asymptomatic people during this incubation period are shedding virus that accounts for the huge contagiousness of the virus, of this particular virus. That's because the virus has shut down the innate immune system, and they're the first responders. Those are the guys in the ambulances and so forth, and so the first responders don't respond. And therefore, the virus just replicates unimpeded. And there's one more aspect about this whole process is that the reason there are no symptoms, so that you don't know that you're infected, and you're walking around, and if you're a college kid, you're going to the fraternity house and drinking beer and having a great time with all your friends. Or if you're down here in Florida, for example, you're out on the beach, and you're asymptomatic, feel fine, there's no problem. And that's because the symptoms of the infection are not due to the virus. The virus itself is it's a stealth bomb. It doesn't alert the host that it's there, except when it stimulates the innate immune system on the part of the host, or the adaptive immune system, to respond. And so all of these symptoms of fever and chills and cough and all that stuff that we associated with a cold, an upper respiratory viral infection and so forth, that's due to the immune response to the presence of the virus. And since there's no immune response because we've stopped it all, because we can't make interferon, then there's no symptoms. And so that's all really laid out in this prescient review article by Eleanor Fish. And that's why I think everybody should check out that paper and read it as much as you can. It gets pretty complicated and it's pretty condensed, but the essence is what I just said. The next thing that happened with Eleanor is that in 2003, when they were having 8,000 people worldwide got SARS-CoV-1, and there was about a 10% mortality rate. Exactly the same kind of symptoms with CoV-2, and that is the patients were presenting with shortness of breath, and they ended up in the ICU on ventilators and so forth. Eleanor did a short clinical study as a pilot study to see whether or not interferon given to patients would actually benefit them. And she basically showed that. It decreased the amount of virus that they could measure. It decreased the time that patients had hypoxia and shortness of breath. And it shortened the time that they were hospitalized. But it was only a few patients. It wasn't a so-called randomized controlled trial. It's abbreviated now in the literature as an RCT. Just to confuse you, you have to go back to your glossary to figure this all out. But anyway, she published that in the Journal of the American Medical Association, JAMA, which is one of the most well-known clinical journals. Basically, that was 2003, so that was 17 years ago. And you say to yourself, so what was everybody doing in the meantime? Then they had MERS that came on in 2012, 13, 14, along in there. And still, people didn't, they thought, well, glad that's over with. We don't have to worry about that anymore. And so now we're in the fix that we're in today. And so this is not going to go away. I mean, there's going to be more of these coronaviruses. And so we have to get our act together and stockpile and do all the things that the politicians are not doing. Then if you fast forward 2020, in May 15th of this year, there was a very, very important article published in Science, which is well read by all the scientists, but not necessarily well read by all the clinicians, we'll see. And I've been trying to beat the drums on this particular paper as well, because this paper was done by a whole team of French investigators. What they did was they did a cross-sectional study of patients that were infected with COVID-2. And they had, I think, 50 patients, and they separated them into three different groups. They had 15 patients that they, by their symptoms and so forth, they labeled them as mild or moderate. And then 17 patients that with severe coronavirus infection. And then 18 patients with very critical. I mean, these are people in the ICU probably on ventilators and so forth. So what they did was they which I think is just wonderful and beautiful, because they took all of the science that has been created into understanding the immune system and that have happened in the last 50, 60 years, actually, that I talk about in my book, Molecular Immunity, where I detail. The reason it's called molecular immunity is that we spent the last 50 years understanding what the molecules are in the immune system that are really doing the tricks. What these people did it's a whole team of French investigators, and the title of their article is Impaired Type 1 Interferon Activity and Inflammatory Responses in Severe COVID-19 Patients. And what they found, what they talked about in their introduction of this paper was, they say it's sort of strange because the patients are presenting, and they have sort of upper respiratory infection symptoms, and they're symptomatic, but definitely not really terribly sick. But then after about 9 to 12 days, they start to really go downhill fast, and particularly in the respiratory system. And so they hypothesized that this had to be due to the fact that there was an imbalance in the innate immune system, and also an imbalance in the adaptive immune system. And they correctly said, the symptoms in this disease are due to the immune response, not due to the virus. And so they did all kinds of assays. on these 50 patients. And it's a cross-sectional study. It's not that means that it was a slice of life. It wasn't a longitudinal study where they followed patients, down from the beginning to the end of their illness. So it's cross-sectional. And that's important because of the findings, actually. And they looked at all the cells. And now in immunology, we have these fabulous machines whereby we can analyze individual cells. We can look at the outside of all the molecules on the outside of the cell. We can look at all the molecules, or a lot of them, on the inside of the cells. We've categorized all these cells depending on what molecules they express on the outside and on the inside, and what they can make, and so forth. And so they looked at all of that, and they found that there was no any real great change. Well, there were decreased numbers of T cells and natural killer cells. And as the severity of the symptoms changed from these different categories, the T cell counts and the NK cell counts went down. And there was also a relative increase in the number of B cells and macrophages, and then also an increase in the number of plasmablasts. Those are the cells that are cranking out antibodies. And the T cells that were there had on their cell surface a bunch of activated T cell molecules, activated molecules. And they also had molecules on their cell surface, for example, PD-1 that I talked about in the cancer immunotherapy end of things, and the co-inhibitory molecules. PD-1 has become a symbol or a characteristic of T cells that they've now termed that they're exhausted. Sort of like as you get out of the gym, you can't really hardly walk out through the door. That's an exhausted T cell. And there were molecules that they could detect on the outside as well as the inside that indicated that the T cells were undergoing cell death, programmed cell death, or what they call apoptosis. So what they found then so that's just the cells that we can do really very, very well nowadays. 20 years ago, we hadn't even discovered these molecules. So they found that as the symptoms in these different categories were worse and worse, there was fewer and fewer interferons that they could measure. For example, first of all, interferon beta, which is the one that's made by the macrophages and the dendritic cells, basically was unmeasurable. They couldn't find any. And the alpha-2, which is one of the most common ones that is measured, was markedly down. So they also measured interferon activity from these cells, and meaning that they measured to see whether or not it would interfere with viral replication, and that was down. And it was it was further decreased in the most severely ill patients. When they added interferon to the cells, they could show that they could have a brisk interferon response. So that meant the problem, just like Eleanor Fish had said, the problem here was there was no interferon produced. But if it was produced, or if you gave it to them therapeutically, it would help because there was an interferon response that they could measure. Now, the other thing that they measured was as the patients got sicker and sicker in these three different categories, the virus concentration in the body went up and up and up. So there's low interferon or no interferon and increased replication of the virus. And then you've got virus all over the place. And now the acquired immune system begins to kick itself in. And what they found was the sicker the patient, the higher the levels of interleukin 6, particularly, that they could measure. Interleukin 6 and TNF alpha. They also looked at interleukin 1 that I've said is the third of or the first or the second of the pro-inflammatory cytokines, and they didn't find too dramatic differences in the interleukin 1 end of things, but they did find there were marked elevations of TNF and interleukin 6. And so all of these findings in this cross-sectional study and how they end up their paper is that they say, well, you know, what we ought to be doing is probably giving these patients interferon on the one hand, and we should be then giving them cytokine antagonists, particularly for interleukin 6 and for TNF alpha. And I've looked online, and I can, you know, and you can't find any real data out there with regard to cytokine antagonists yet, but there's several clinical trials that are ongoing and that are right now. And I sort of feel that, you know, the cancer people, the oncologists that were using these CAR T cells, the chimeric antigen receptor T cells, that then activated the so-called cytokine storm. So they had these patients that were really sick, number one. Number two, they caused it. So there was nobody was going to do a clinical trial, for God's sakes. They just reached off the shelf and gave them IL-6 antagonists, and the patients got better within 24 hours, and they were going to die. So I'm saying to myself, as a scientist, formerly a clinician scientist, you don't have to wait for a clinical trial, and you shouldn't wait for a clinical trial. You should just be treating these patients, because this is a pandemic. People are dying. But that's my conclusion on that end of the story. So now there's one other paper that I've become excited about recently, and that's a paper that came out in Cell. It's published on June 25th, 2020. So just, you know, a month ago now, because this is the 21st of July as we talk. And this one is entitled Targets of T-cell Responses to SARS-CoV-2 in Humans with COVID-19 Disease and also Unexposed Individuals. So what they're saying by the title of this paper is that they're trying to find out what parts of the virus activates T cells through their T cell antigen receptor. And they've been studying these kinds of things for at least a decade, maybe more. The senior author is a fellow by the name of Alessandro Sette, and he's in La Jolla, California, at the Immunology Institute, La Jolla Immunology Institute. And what they did, they've got it worked out now so that they can sequence the virus, find out the nucleotide sequence of the virus, and on the basis of that, they have predictive programs that can tell them whether or not there are peptides that could be coded for by the sequence of the DNA that might be able to be bind to and be presented by both the major histocompatibility complex class 1 molecules, which would activate CD8 T cells, and the class 2 molecules that would activate CD4 positive T cells. And so they've done that. They've done a study where they compared 10 COVID-2 patients who were sick and had recovered. And then they had 10 individuals who were normal and never been sick and never been infected with COVID-2. And so what they basically found was that they found that they found peptides from the virus that would activate all 100% of the CD4 positive T cells in 100% of the patients, I mean. And in 70% of the patients, they found peptides that would activate CD8 positive T cells. And the other thing is that all of the vaccines this is important and basically what they say in their paper is that we're trying to establish a baseline of data and what happens during a normal infection because that's important for what we look for in our vaccines, to see what kind of an immune response we would hope to get. And all the vaccines basically are focusing down on the so-called spike protein of the virus, which gives the name of all the little spikes coming off the virus, because that's what binds to the receptors on the cells, and that's how the virus gets in. So if they can get an antibody response to those spikes, then that should be, and it has been shown to be, a neutralizing antibody, and that is it will neutralize the virus. It won't be able to get to infect the cells, and that should be a good vaccine. So Sette found that they did find peptides from the spike protein that would activate T cells, but they also found major structural proteins that the virus has, that codes for, that make up the body of the virus, and they found peptides there that would activate from these convalescent people, that they had CD4 positive T cells and CD8 positive T cells. Now, the other thing that they found was, and this is really important, they found that when they looked at peptides that were the same peptides in COVID-2, but now the peptides had the sequence of the coronaviruses. There's several of them that cause the common cold. They found evidence of reactivity in the COVID-2 patients. So that means that there's cross-reactivity to other coronaviruses, which is not unusual. I mean, that happens in most viral, all these viruses they're sort of like mafia. The mafia that has a big family and lots of individuals in there. And so, but the importance of that, I think, and they think so too, is that it could very well be that these patients that are infected with COVID-2, that are either asymptomatic always or just mildly symptomatic, there could very well be cross-reactivity, cross-immune reactivity from previous cold virus infections that are responding to the COVID-2 infection, and so that's why they're not getting really sick. They're not getting really sick. They can make interferon, and they can deal with the virus right away rather than let it go willy-nilly, essentially. So there's good science going on in the world, all over the world, in China as well as Toronto and as well as La Jolla, and I think that there's hope. There's also some things, I think, with regard to this virus, and because the other members of the family, they're cold viruses, the immunity, and this is one of the big questions that you'll read about in the newspapers and so forth. The question is, okay, let's say you get infected and you deal with the infection, and now are you immune, number one, to reinfection? And number two, if so, how long does it last? And it could very well be that coronaviruses do not cause long-lasting immunity, and so that you can become reinfected. And I think that's an open question still. We'll see what happens in the mix of things. But HIV, for example, there's two things about that. HIV, there are no individuals, except I think one in 25, 30 years, that cures themselves. And you don't have any people that will be able to make the virus go away all by itself, all by themselves. So there we are, and what can I say? The beat goes on, and I'll stop there.